Micro. It micro. And the answer, the correct answer is microtubulus. Yeah. Yeah. Tại vì em nhớ là trong cái phần học cái này thì nó ở trong cái phần là
Okay,
microtubulus.
so in
Ngay
my slides, when I talk about the
Vâng,
Stilla and
đúng rồi.
flagellus, they are in the parts of microtubulars, right? So the Stilla and flagellus are comprised from microtubular. Okay, may I ask the second students? No, the third one.
Yes.
Kneelings. Okay. So the top 10 students, please cap this. I don't know, this report, because some case indicate that I lost my picture. Okay. I will give you a file or a tables. Classify the student who received the 0.25, and you will check this again, double check with me. So can you explain the question number 4 or to your
number
class?
4. Number 4, I wrong.
Are you wrong?
Yes.
Okay, sorry. BI trials 026. So 9.
Sorry, I have some technical problem. I cannot hear that. Can you explain?
Sorry, could you please explain the question number 4, 5, and 6 to your friend?
Can I see this question again?
This question is on my share screen. Question number 4.
4. So I can see anything. I can't see your screen. Oh no. My Zoom is problem. I need to get out. Sorry, my Zoom has some problem.
So okay, Nguyễn Anh Minh, BI trials 291. Are you here? Nguyễn Anh Minh, BI trials 291. Or he not here. So Nhi Ling's BL 12339. Can you explain the questions? So for the number 4, I will explain to your class which cells component associated with motility is of course cytoskeletons. And number 5, Nhi Ling's. For the number 4, it is a cytoskeleton is the combined of centriole, centrosome, microtubular is all the component of cytoskeleton. So when you choose the cytoskeletons, you make all the other answers right. Okay. And Nhi Ling's number 5, please.
Yes.
Question number 5.
Yes. Question number 5 is five is in the fiber.
The thickness fiber.
Thickness. Yes. Um. It default right. It, because it has long about 25 nanometer, while other is just about 8 and 10
Okay,
nanometer.
so remembers the size this kind of fiber inside the skeletons. We have the biggest ones is microtubulars. So pay attention. We have intermediates. It means they are in betweens. Remember, they are in between. So I mean they have the size in betweens actin filaments and also the microtubulars. So the biggest one is microtubulars. The second one intermediate filaments, and the last one, the thinnest one, is actin filaments. Okay. So Nguyễn Anh, I don't know.
Thế cho em hỏi tại sao
BHL.
là thickness là kiểu thickness em bị độ dày ấy?
Đúng rồi, nó to nhất và nó cũng dày nhất luôn.
Không, em tưởng đấy là đuôi EST nhưng mà cô là ESS ạ.
Sao cơ?
Which is the thickness em tưởng cái này đang là dày nhất ấy. Thì tại sao nó lại là đuôi là ESS ạ?
Sorry. sorry.
Vâng.
Yeah, there's a typing mistake here. Nguyễn Anh, no. Nguyễn Anh Minh. Nguyễn Anh Minh, you randomize all the questions or only number 4? Sorry, take a lot of time in this game. So, okay, Linh, can you explain for me the next question also?
Question 6.
Yeah, number 6. We don't see here.
Parfait. Yes. Metaphase is a third of mitosis. The process that separate genetic material carry in nuclear parent cell into yes.
So, okay, for this question is my appears in your final exams. And as I said, that's for the parts of cell divisions. I will give you some kinds of picture and you have to guess what phase it is. And remember, that's 2 phases that's easy to recognize is metaphase and the anaphase. So for the metaphase, M's present for the meters. So easy to remember. So in the meters, so you can see that's all the chromosomes here, they are lined up in one line in the meter of the cell, so they are metaphase. And when they are separate, they goes away from each other, we have the anaphase. And in the telophase, they started to form 2 cells, okay? It's the easiest way to remember of the cell divisions. And what about the question number 7? Okay, I don't know, but we have very high percentage of students who cannot give the answers. That's because you don't have a lot of times, or maybe you have problem with your internet connections, or you don't understand the questions. the rate of the student who give no answer is occupies maybe 20 to 25%. I think it's very huge. So because we don't have time, so I will explain very fast about the question number 7, 8, and 9. All right, and the accuracy rate of this question is quite low. And actually, I really expect the students who who give the right answers of the all the questions. Students number ones give me the answers, but he have very smart way to avoid my requirements. Okay, so I will give him 0.25. That's okay. For the question number 7, what are the thin tails light fibers surrounding their cytochromatis? Very high accuracy rates. Okay, they are spindle fibers. Spindle fibers we go to attack the kinetochore of the cytochromatis and pull them back into the two or opposite poles. Okay, and number 8. This condition is necessary for a cell to qualify through the Z2 checkpoint. And with almost of the questions relating to the checkpoint, remember for me the DNAs. So the job of the checkpoint is most of the case to ensure that our DNA is okay. There is no damage on the DNA. So in these 4 answers, you may find this only the answer B have the complete and accurate DNA replications, because the Z2 is right after the S phase. In the S phase, we have the synthesis of the DNAs, and or in the G2, they are in the growth phase and they try to produce some other material for the cell divisions. And for the G2, they also need to check whether the DNA is okay or not. So they need to check these DNA replications is complete or not, and is there any damage in the DNAs. And the question number 9, cyclins is associated with this mitosis. I don't know what it cytolysis is a treat. I just think about these words, but I don't know what is the cytolysis at all. So cyclin is associated with mitosis. Also cyclin the CDKs, remember about the cells cycle regulators. Okay, they are positive cell cycle regulator. Cyclin plus the CDKs. Revise a little bit of my lectures, because we have 2 more lessons to finish this course, and after that we have a one tutorial class. In that class, I will remind you all of the information in this course, and also we practice the multiple choice question a lot. But before you go to this tutorial class, please revise yourselves. Okay, so we move to the lesson today's. Let me share screen too. So in my previous lectures, I talked a lot about the hypothesis and also the cancels. And today we will go discover what are these, okay? For hypothesis, as I tell you that they are the program that the cells need to kill themselves. If they don't receive any cell growth, like in the lecture about the cell communications, when the cell receives no signal from the external environment surrounding them, the survival signal, they will go or they will kill themselves by hypothesis. And also during the cell cycles, if it at any checkpoint we discover some DNA damage, the damage in the DNAs, but is unfixable, the cell needs to go to the hypothesis also. So what are these? The hypothesis. The hypothesis, they have another names like the program's cell days and also the cellular suicide. So program's cell days is mean là chết theo chu trình because they are programmed to date. Oh, sorry. The definition of hypothesis, there are generous and convenient ways to remove cells that should no longer be part of the organisms. Because during our developments, there are some cells need to be deleted. You know which process that we need to delete the cells. And second, some cells are abnormal and could hurt the rest of the organisms if they survive, such as cells with the virus infections or DNA damage. Okay, if the cell is infected by the virus or bacteria, or there are some mutations in the DNA. We don't want that kinds of mutation go to the next cell's descendants. We need to remove them. And the cells in adult organisms may be eliminated to maintain the balance, to make the ways for the new cell or remove the cell needed only for temporary tasks. Sorry, is no explanation for this, because when you grow, some cells need to be died, and the younger cells will take the job of these kinds old cell. So the first one, program cell days hypothesis, is a normal path of development. So you can see in these pictures, we have a section of the interdigital web. So the cell days, and they staining the nuclei, and you can see that the cell dates have the characteristic of the hypothesis because they have numbers of the nuclei here. They are disintegrated. And this process occurs in all sorts of vertebrate species that have a finger or toe. So during the development of the embryo of the vertebrate species, if we want to make our finger or toes here in the poles of the embryonics, there is hypothesis here, hypothesis here, hypothesis here to separate our fingers and our toes. And that is the part of the development, neoplastic developments. And the second are the hypothesis can eliminate the infected or cancerous cells. I remind you a little bit about the P53, remember P53? P53, the tumor suppressor genes. And when is any checkpoint that we discover the DNA damage? My previous lecture. If DNA damage, we figure out some DNA damage, the P53 will increase, and he have 3 jobs, right? First, they need to stop the cell cycle. One, they stop the cell cycles to take time for it to fix the DNA damage. So the second step, they will increase the levels of the DNAs of polymerase that can fix the DNA damage. DNA is also one kind of enzyme here in the industry. So if it's unfixable, right, it's unfixable, they will activate the hypothesis. This cell with DNA damage need to be died, so we can prevent the risk of cancers in our bodies, right? If the cells do not allow follows the P53 command, they not die anymore. So the DNA damage can pass through the next descendants of the cell, next generations, and they are pre-cancerous cells. And if they keep dividing many more times, they could lead to cancer. So it's really, apoptosis is quite important to our bodies. They prevent the cancers, they create our toes and our fingers, and what else? Here. They are the keys to immune functions. The apoptosis plays an essential role in the development and maintains of the healthy immune system. I remembers about the communications lectures when I told you that two immune cells, they are contacts with each other by no distance. Remembers when we have a macrophage here, macrophage, bạch cầu ạ. And after they digest the virus, for example, they will send one small piece, antigens into the MHC receptor of their surface, and a T cell will come and recognize these antigens, and they will activate the reticular cell to help the macrophage to kill the antigen, to kill the pathogens, and also they activate the B cells to produce the antibodies. Remember, and also the B memories to store information of these pathogens for the next time they invade our bodies. So, what is the roles of apoptosis in this process? When the B and the T cell, the B cell and the T cells are first produced, they need to ensure that they are react against our body cell component or not. So if they are first produced and instead of attack the pathogens, they are attacks our body's own cell, we have the troubles. Yes, if they are attacks, they need to be died, because if not, they do not go to the apoptosis. We have a problem with autoimmune conditions. Do you know what is the autoimmune conditions?
The immune
you ever heard
system
that?
fight against the body cell.
Yes, when the immune system
Recognize the body cells as foreign matters.
No, they always recognize our body cells. The problem is they don't recognize our body cells. Every cells for them are enemies. So they can find the pathogens, they find the virus, they find the bacteria, and they also find our body's own cells. So that's why they are apoptosis. They are plays an important role in allowing the immune system to turn off its respond to the pathogens and turn up the response to maintain the homeostasis in the immune system. So autoimmune conditions or autoimmune disease, they are one disease that our immune system find our bodies. Do you know very famous ones? Lupus bandar. Can you search for lupus bandar or for the autoimmune conditions? So there's very stationary that there's a child 5 years old. He's my neighbors, and he just passed away because of the autoimmune condition, autoimmune disease. That's the immune system of our own bodies attacks ourselves. Very dangerous. So that's why the hypothesis is quite important for us. And the next, after they are, when the pathogen is or detected after the B killer and the B cells or removes all the pathogens from our bodies, the immune cells need to be died because we don't need a lot of them inside our body. It's take energies, it take everything from our body. So they need to be removed and they need to die following the hypothesis. Okay. First, to the immune system, first test if they are find our body cells instead of the pathogens. And the second, after they are, or after pathogens clear, we need to remove also the immune cells because we don't need numerous of them inside our bodies. And for the next, we have two comparisons, the apoptosis and the necrosis. So we have another kind of the cellular damage. This is necrosis, and they have different appearance than apoptosis. And here we have the necrosis on the left and the apoptosis on the right. So the apoptosis is the process which a cell designed to kill itself because of DNA damage, because they withdraw the essential growth factor and nutrition. I remember this. In my cell communications, I taught that if the cell do not receive any growth factor, receive any signal from the environment, they will or die through the apoptosis. And the cell is not detachment from the substrates, or they are attacked by the cytotoxic lymphocytes for many reasons. So these are the reasons. Thus, we need to go through the apoptosis. But for the necrosis, it's just the cells have the injuries. Or for them, when we have been accident that we cut our fingers, this is also the injury of the cell also. And for the apoptosis, the nucleus will be fragmentations into small pieces, and all the parts of the bodies will be packaged into small vesicles like this. This is the vesicle, small vesicles. And then the phagocytes, they will be engulfed by other cells to reuse these kinds of the fragments. And there's no inflammatory response. Inflammatory lymphoma. Do you know inflammatory? When you have some problem with your bodies, when you have some pathogen attached, we have the inflammatory. That is the reaction of our immune system. The phenomenon signal of inflammatory is when you got fever, right? Fever is the good signals. Is the sign that your body is fighting against the pathogens. But as for the autoimmune disease, because they don't know what is the pathogens, what is your own body self, the immune disease or patients, they may get inflammatory effect, they may get fevers without any attacks from the pathogens. That's a problem of the autoimmune disease. So what about the necrosis? For the necrosis, the cell will be swelled, burst, and we have the inflammatory response. That's what different between them. And also I have a term that's the autolysis. So when we talk about the apoptosis, it means that a cell kills itself. But for the autolysis, it means an enzyme kills a cell. And also they are different from senescence. the difference from senescence. So apoptosis is a process with cell designed to kill the cell, but the senescence is an irreversible arrest of the cell proliferations while the cell maintain the metabolic functions, and often associated with the cellular aging. Both processes are induced when the cell sense that the DNA in the cell is damaged, but the hallmarks of apoptosis is the sequential cellular event lead to programmed cell death. And these two events are not related, and they have a distinctive biological pathways. I don't teach you about the senescence, but I teach only about the apoptosis. And the senescence, they are shown to be resistant to apoptosis. They're different. Okay, so what happens with the nucleus during the apoptosis? So the cell structures change during the apoptosis. You can see the left panels to show a normal cell. So the nucleus, they are predominant organelles. They are the biggest organelles in the center of the cells. And in the right, you can see the apoptosis cells. The golden arrows indicate the condensed nuclear fragments. So there is the fragmentation of the nucleus during the apoptosis. And here's the we can detect the DNA fragmentations by the electrophoresis. Electrophoresis experiments. So we extract the DNA, the total DNA here. The total DNA here. He puts them in the wells, and then the DNA will go follow the electricity from the up to the bottom here. And you can see that if the DNA is normal DNA, the total DNAs, they are really, really big. They will stuck in the wells here. But the small size of the DNA, this is the small piece. They will go through the gels, and we can see them like this. And for now, we have in the well number 1 and number 2, we have the normal cells. And the well number 4 and number 3 and 4 is might be apoptosis, right? Apoptosis happens. Here, this is mechanisms, or all of the mechanism is quite complicated. So mechanisms of apoptosis is a little bit complicated. So here are the step 1s. I call them the step 1s. Remember cell communications when the ligands go to meet the receptor is the first step of cell communications, and also the first step here in the mechanisms of apoptosis. So lies more signaling between the cells. We have two pathways, extrinsic and the intrinsic pathway in apoptosis. And first, about the extrinsic pathway, and the first step of the extrinsic pathway, they start with a signal molecules binding to a receptors on the outside of the cell membrane. Here, the ligands will go to bind the DATE receptor, because what is DATE? Because we result in the cell DATE, so we call them the DATE receptor. So two common types of chemicals messenger that trigger the extrinsic pathway to apoptosis, they are FAS and TRIR. Here. So in the step number 2, in response to change in the intracellular domains of trace and fast receptors, a protein inside the cell we call the FAID also chain. This is the step number 2. The protein, you see the green protein here, FADDs. FADDs also chains. FADDs are sorry, the names of them is the fast. Remember the fast receptor here. Fast are associated. Fast associated date domains. Fast associated date domain proteins also change in the step number 2, and once it has been activated by the change of the receptor, it interacts with 2 additional proteins which go to start the process of the cell death, the procaspase 8 and the procaspase 9. So in the step number 3, the procaspase 8 and procaspase 10 are inactivated proteins until they are interact with the activated phase. So this is number 3. Number 3, when the procaspase are associated with the fast active protein in green here, they will be activated and become the caspase 8 and caspase 9. So pro become caspase. This is step number 3. And in the step number 4, we have another inactive molecules we call the BID, the BID. For the BID transformed into the T-BID here, and just because the caspase 8 and caspase 9 cleave it, this part of the BID protein to become the T-BID. Caspase 8 and caspase 9 cleave or cut this part, and they become active. The T-BID is the active form, and they move to the mitochondria. So this is step number 4. When caspase 8 and caspase 10 cut this part of the BID protein and they become active, and they will go to move to the mitochondria. So the step number 1, 2, 3, and 4, they are unique to the extrinsic pathways. But after they relieve the T-BID and they will go to the mitochondria, they will activate the BAX and BAX protein here. We call them, they share information with the intrinsic pathway. And what about the intrinsic pathway? For the step number 1, the intrinsic pathway here, this step number 1 here. I will change the color. I talk about the intrinsic pathway, and this is number 1 of the intrinsic pathway. The intrinsic pathways to apotheosis is the trigger by stress or damage or to the cells. So it could be lead to the apoptosis, maybe is a DNA damage, maybe the oxygen deprivations, and other stresses that impairs the cell's ability to function. They are the reasons of the apoptosis and intrinsic pathway. So in response to these damage or stresses, the cell decides it continues. Extrinsics might be dangerous or costly to the organisms like heroin, then they activate a setup protein we call the BH3 only proteins. So in the first step, they activate the BH3 only protein here. On step number 2, the BH3 only proteins, they are class of protein including several pro and anti-apoptosis proteins. Apoptosis can be encouraged or discouraged depending on which BH3 only protein that activates or expresses. The pro-apoptosis BH3 only protein, they will activate the BAX, BAX, and BAK protein. This is the step number 2 of the intrinsic pathway, where the BAX3 only protein activated in the step number 1, and now they activated the BAKs and BAX protein in the step number 2. So the TBAs in the extrinsic pathway also go to activate the BAX and the BAKs proteins. Activated BAX and BAKs cause condition we call the MOMP. MO, you see here is the MOMP conditions. The MOMPs is they are stand for mitochondrial outer membranes, permeabilities. So it affects the permeabilities of mitochondria. These conditions, by activate the BAX and the BAKs, is will cause conditions mitochondrial outer membrane permeabilities. So the MOF is considered the points of no returns for apoptosis. Once conditions happens, when this MOF happened, the apoptosis is activated. So now we cannot stop them. We call them the point of no returns. And this step is leading to MOF can be started in the track of inhibitor molecules, but one MOF has been achieved, the cell will complete the DATE process, the point of no return with the MOF. And the MOF plays its key roles in apoptosis by allowing the release of the cytochrome C into the cytoplasm. So when the BAX and the BAKs, normally they have this BCL protein here, they are in connect with the BAX and BAKs. So when they have been inactive by the BH3 proteins or by the TB proteins, they relieve the BAX and BAKs. And this BAX and BAKs, they will form a channels here. They will form the small channels here. In our case, BAX and BA, sorry, it is BAX and BAK. So in our case, they are in connect with. So the red one is maybe the BCL. And the green one, they are BAX and BAKs. In our case, they love, the BCL is love this BAX and BAKs, but when activate the apoptosis, they have been inhibited by the TBX or by BH3 only proteins, and the BAX and BAK will activate, and BAX and BAKs form the small channels here to affect the mitochondrial outer membrane permeabilities. They form the channels and they allow the cytochrome C here, relieve into the cytoplasm. That is the step number 3, activate the MOF. And in the step number 4, under normal circumstances, the cytochrome C, they plays a key role in mitochondrial electron transport chains. So when there is escape of the mitochondria, they act as the signaling molecule in the cell cytoplasm. The cytochrome C in the cell cytoplasm, they bring the informations of the amino surrounding the apoptosome. They will go to bind to the apoptosome. They are a complex of protein that perform the final step to begin the cellular breakdown. In the step number 4, the cytochrome C relieve into cytoplasm, form a apotosomes. And the step number 5, the apotosomes, one is formed, they turn the caspase nemanase. They activate the caspase nemanase. Apotosome activate the caspase. And with the activation of the caspase, you remember 8 and 10 here, caspase is able to trigger further through the cells by the activase. They activate the caspase 3 and caspase 7 in the step number 6. And here remember that in the extrinsic pathway, the caspase 8 and caspase 10 also activate the caspase 7 and the caspase 3. So in the step number 7, one activates the caspase 3 and caspase 7 begin to break down all the materials. So this is 7th step or the final step. For example, the caspase 3 will condense and break down the cell DNAs. And for the caspase 7, they will break down other parts of the cells or cellular materials. That's all the mechanisms. And I will show you videos, and you can see that's how the caspase will be activated and how the caspase will go to break down all the materials inside our cell. Okay.
Apoptosis is an important cellular process that allows cells to die in a programmed fashion. Apoptosis plays an essential role in growth and development, such as in the womb, where the fetal hand starts out as webbed, and fingers are formed through the programmed death of the cells in the web spaces. Apoptosis also plays an important role in removing faulty cells. For example, pathological apoptosis may be induced if cellular DNA is damaged beyond repair. In apoptosis, the cell is broken down from within by proteins called caspases. For apoptosis to occur, these caspases first need to be activated. Caspase activation can happen via 2 distinct pathways, called the extrinsic and intrinsic pathways. The first pathway is referred to as the extrinsic pathway because the initial signal comes from outside the cell. This pathway is often initiated by other cells, commonly by subsets of T lymphocytes. These lymphocytes have a surface molecule known as Fas ligand, or Fas-L for short. The extrinsic pathway is initiated when Fas-L binds to Fas receptors on the surface of the targeted cell. This sets off a chain of intracellular events that will ultimately result in apoptosis. The sequence is mediated by a Fas-associated death domain, or FAD. In the final step of the extrinsic pathway, caspases activate each other in a self-amplifying process called the caspase cascade. Apoptosis is then initiated as the active caspases begin the breakdown of cellular materials. The caspase cascade acts as a common endpoint for the second apoptotic pathway, known as the intrinsic pathway. As its name suggests, the intrinsic pathway is initiated by. This intrinsic pathway is regulated by maintaining a balance between 2 sets of proteins in the mitochondrial membrane: anti-apoptotic proteins such as BCL2 and BCLX, and pro-apoptotic proteins such as BAX and BAC. In a healthy cell, the anti-apoptotic proteins bind to the pro-apoptotic ones, thereby blocking their action. But if a cell is damaged, or if it stops receiving survival signals, BCL2 and BCLX are blocked in turn. BAX and BAC are then free to punch a series of channels in mitochondria, allowing mitochondrial substances, such as cytochrome C, to leak out into the cytoplasm. The leaked cytochrome C binds to APAF1 proteins to create a compound that then activates the caspase cascade. Apoptosis plays a key role in growth, immune surveillance, and neoplastic development. In all of these processes, there can be abnormalities of too much or too little apoptosis. For instance, cancer cells survive and replicate because they are able to block apoptosis. Understanding the functioning of the extrinsic and intrinsic pathways, as well as of the caspase cascade, allows us to design targeted therapies based on better regulation of apoptosis.
So at the end of the video, so I we have some application in this case. For examples, I'm a scientist who find out the compound for the cancer treatments. Okay, so it's my job now. For examples, if I find one compound that can inhibit the growth of the cancer cells, I want to test whether this compound is kill the cancer cells or through apoptosis or not. How can I test? Based on this kinds of theories, how can I evaluate this cancer cell is dead through the apoptosis or not? Do you have any suggestions? Anything that I can test?
Could you repeat the question, please?
So the question is, I'm trying to find out the compound that can inhibit the cancers, and I want to ensure that my compound that can kill the cancer through the apoptosis. How can I test that these cancers are dying through the apoptosis or not?
I think we can measure
Can I evaluate any part of the cells? Cái phần này nhá, giả sử như là công việc của cô bây giờ cô đang tìm rất là nhiều chất để chữa rất nhiều bệnh chẳng hạn, và cô có một project về tìm các chất để chữa ung thư nhá. Khi mà cô đã tìm ra được rồi đúng không, thì bây giờ khi cô đã tìm ra được cái chất đó chữa ung thư, tức là nó đã inhibit cái tế bào ung thư rồi, mà bây giờ cô muốn test xem là cái chất đó nó diệt tế bào ung thư theo cái chu trình apoptosis hay không, thì cô có thể dùng các yếu tố nào để cô test cái này nhỉ?
I think we can measure the concentration of the caspase protein.
Caspase, right?
Caspase.
So evaluate.
So
evaluate.
if we
caspase level, the increase or decrease, right?
Yes.
So which criteria?
I think it can be a cytochrome C2.
Cytochrome C2. Yes, might be.
I think that any step that beside the MOMP condition that cannot return will lead to the apoptosis. So if this step happens, then the cell might be killed by the apoptosis.
Mm-hmm. So process related to the MOMP conditions. So it's also measure the level of cytochrome C in the cytoplasm or not. It's not really easy to perform. So next, what criteria that I can used to test the hypothesis of the cells? Remember from these videos, can you see that the BCLs, they are block the BAX and BAKs. Remember? When they are disintegrated, when they are separated, we have apoptosis because the BAX and the BAX and BAKs will activate the MOPS conditions. Can we use this signal to test the apoptosis? Find out that do we have BCL and the BAKs because now they are separated. Can we use them? And the next remembers a slide that I showed you that the pictures, we stain the nucleus and we find out that the apoptosis cell have the fragmentation of the DNAs. They don't have the normal DNA, so we can test also the DNAs, right? And also we can extract the DNAs. Remember the picture about the electrophoresis? Điện Z proteins. So any kinds of tests here, we can choose this, point out 3 or 4 here to ensure that these kinds of cells are undergo the hypothesis. So when we learn about the theories in biology, we need to apply in the research and with all the other major cells. When you study the theories, you need to think that's how could we apply in the research studies, okay? And also it might be because I'm working is finding the compound related to the cancer research, right? I'm finding the compounds with called inhibit the cancer cells. So it might be with that the level of p53. Can we? So we can test the level of proteins, we can test the level of DNAs by numbers of experiments to find out that the process occurs or not. Okay, even the when we see the DNA, that we see the shape, not the shape, or when we evaluate the proteins, or when we evaluate the fragmentation of DNAs, this means we evaluate the mechanisms. Is all good for our studies. So we will have the break times before we move to the second part about the cancers. Okay, we will be back at I don't know, it's 11:10. Yes, 11:10 more. You have any questions about the hypothesis?
No questions.
Yes. About the mechanism is quite complicated, but when you see these videos, I think it's more clear. Okay. So watch this at home again. So we have a break time now. We come
Yep.
back to the lessons.
Yeah.
And in the part 2, we're going to discover what is a cancer. Okay, as I remind you something about the cell cycles, where all the normal cells, they are living most of the times in the interface, right? But here the mitosis with the cancer cell, they are exceptional. They are exceptions because most of the time the cancer cells in the mitosis stage. And what else? We have a several checkpoint here, and the checkpoint here. If the cell with the damage pass through this kind of checkpoint, they might be the pro-cancerous cells, and with several types of division, it will lead to the cancer. So what is the cancer? The cancer cells is basically a disease of the uncontrolled cell division that we are the cell division unstoppable. We have the cancers. So okay, my job is working with the cells. I culture the cells outside your bodies. Outside, I culture the cell in the dish like this, and the multiply in the cancer cells. We don't need to adding any growth factors. So here with the normal cells, when I culture the normal cells, I need to add the growth factors. Our cell will divide without growth factor. There's no divisions like in the cell communications lectures. I so use a diagram that's a picture that's when a cell receives no information from the external environment, they will be died. And this in the case if without growth factors, the cell with no division, and in the end they will die. But with the cancer cell, we don't need to add any growth factor, just keep culture. That's provide the nutrients, oxygens, appropriate temperatures that they can grow. They can divide unstoppable. They are immortal cells. And for the normal cell, we can keep the culture from the 40 to the 60 times, but the normal cells is in limit. We can keep culture anytime we want. But in general, for me, I perform a research on the cancer cells, so we can stop at the 100 times, because if we subculture a lot, this may be we change the morphology, we change the information of the cell, so we can trust. But in general, in theories, we can cultures so many times of them, they are immortal. Any patches that we want. So what is the here? They ignore the signal that should cause them to stop dividing. As we ask any signal to like remember when P53 the sending informations that you need to be died because you contain some DNA damage, but the cancer cell will ignore all the signal from the bodies. Okay, so that's why we have the cancer, why we have tumor, right? Because our body cannot control them. So the cancer cells, they can replicate immortalities. This means that they can divide many more times than the normal cell. For a normal cell, it's 40 to 60 times rounds of divisions before they lose the capacity to divide or to grow out, and eventually they would die after 40 to 60 times. But the cancer cells, they can divide many more times than this, largely because they express an enzyme we call the telomerase. Remember the telomerase in the lesson about the nucleus? Telomerase. At the end of the chromosome, we have the end of chromosome, we have telomeres, and the enzyme that's maintain the length of the telomeres, we have a telomerase. And the telomerase is highly expressed in the cancer cell instead of the normal ones, because in the normal cell, we don't have a telomerase. Each time we do a cell division, the telomere is shortened, and after 40 to 60 times, they will be died. But for the cancer cell, they have a telomerase, so they can maintain the length of the telomeres. so they can keep divide so many more times. And okay, so the next, they can promote growth of the new blood vessels through a process we call the angiogenesis. Angiogenesis là quá trình tạo mạch trong tiếng Việt. Angiogenesis would give the tumor cells source of oxygen and nutrition. That's for them here, the tumor, they can send the signal to the blood vessels surrounding them, and they can send the signal that's I'm hungry, please feed me. And the blood vessels surrounding them can grow blood vessels into the center of the tumor and provide nutrition and also the oxygen. So that's why for the cancer patient, they are I don't know how could I say this? You can see that it's not really fat people, so most of them is very they are thin people because the tumors use a lot of energy from them, because we need to take the oxygen and nutrition to go to feed the tumor. Okay, and the next, they have the ability to migrate to other parts of the body through the metastasis, quá trình di căn. So that is three characteristics of the cancer cells. So how cancer can develop? We have a hypothesis series of the mutation leading to the cancers. Initial mutations that's related to inactivate of the negative cell cycle regulators. Do you know what is the negative cell cycle regulators? As for the negative cellular regulator, remembers that the proteins p53 and also the RB proteins. And also we have a P21 or RB protein. That's these blood progressions of the cell cycle until certain events or occurs. So, and the next mutation, they are overactivate the positive cell cycle regulator. And it's here, remember CDKs and cyclins, they are positive cellular regulator. I will give you so a little bit about this, reminding you of a cell cycle. In the cell cycles, I draw here a RB proteins, and I have the negative signal here. So they are negative regulator in cell cycles. But here, if we have a CDKs plus cyclins, they are positive. When they go to inhibit the RB, the cell cycle will move to the next phase. And here we have a P53 also. P53 is the negative. So that's why when P53 increase, they can't inhibit, they produce one inhibitor here to inhibit the cyclins and the CDKs. And when they inhibit the cyclin and CDKs, the cell cycle will stop there. So that's we call them P53 RB. They are negative cellular regulators, and for CDK and cyclin, we call them the positive. So in the first step of the initial of the cancer development, we have inactive the negative cell cycle regulator, and in the next step, they overactive the positive cell cycle regulator. So the cell will divide rapidly, right? So in the third mutations, they inactive the genome stability factors. So now they affect the genome, so it's more serious. So additional mutation occur rapidly because in these two steps, the cells started to divide more rapidly. But through the series of divisions. So that means that all the mutation here has been transformed to the next descendant of the cells. And by the end, we have the cancer. So it's not only one cell have been mutation that we have the cancers. It's the process, a very, very long process, and we can't stop the cancer as any step here. But if we can't, we can stop them, all of these steps, we may be the cancers. So it's not easy to occur in our bodies, I'm sure. But at the end, we cannot find them and we have cancers. So here's a graph that will show you the cancer progresses from the local tumor to spread metastasis. So in the first, we have the mutation. In the first step, we have a series of mutations eliminate restraints on the cell divisions. Pay attention, we don't have the cancer with only one mutation. It could be the series of them. We accumulate the mutation, and at the end, we have the cancers. And after we have the cancer cell, we have a tumor growth. They are a group of the tumor cells. They are organized, they are proliferate very quick, and they ensure the angiogenesis to make the blood vessels inside the tumor. Angiogenesis running stomach. And the next step, the intravasation, that the first step that the cancer cells needs to break through the blood vessels to go to the bloodstream. They go to the bloodstream. If they are malignant cells, they can go to blood vessels and lymphatic system. They go through the bloodstream to travel all around their bodies. And the metastasis, when they stay somewhere and they establish the distant organs. So from intravasation to the metastasis, when they travel through the circulation system, through our bloodstream, they might have a lot of difficulty for them. So first, maybe they have a different change in the pH. When they jump into the blood vessel, maybe they cannot survive. And the next is maybe the blood vessel is very tight, is maybe they will broke, or they may be kept by the macrophage. We have the immune system to prevent them to traverse. So a lot of barrier functions in our bodies until they become metastasis. So that is very hard to have mutation. If there's mutation, we will eliminate the mutation. If they become the tumors, it's okay, we can feed them, but they are still benign tumor. And the next one, they jump into our blood vessel, they have so many structure, many obstacle that they need to pass to. And at the end, when we have metastasis, is normally you have no occur in this case. But for mutation to metastasis is very long and very big process to the cancer cells. It's not easy, but it still happens, and we have no cures for when at the final state of the cancer. So what about the mutation? We have a subtype of mutation that's about 10% of human cancer have the hereditary basis. And in most of the case, patient inherits one non-function copy of a tumor suppressor genes. Remember the RB, they are negative cell regulators. Okay, RB here. So example is an RB. Why we have only one RB, one copy of RB non-functions. When we have a mutation in the second time, you may have also the homozygous cell give the rise of tumors. And in this case, need 2 steps, so it's more longer. So some kinds of the cancer have the several types of cancer have the patients have received these kinds of genes from their parents. For example, for the breast cancer, as I remember, just 10 to 15% of these children who receive the genes for the breast cancer may have the possibility to get the breast cancer. And the next one we have these cancer cells is may become the tumors. Tumors, they are abnormal mass of the tissues resulting in uncontrolled divisions. And here we have two types of the tumors, malignant tumor, but we have a cancerous, đây là u ác tính. And the benign tumors, u lành tính. So what is the benign tumor? Benign tumors, they do not spread to other parts of the bodies. Generally localized and small size. They have the slow rate of divisions, and sometimes they have the functions like the normal cells. They do not break out the originating organs, and they're easy to remove by surgery. And for most or some case that the normal or the benign tumors, benign tumors functions like the normal cells, like do you know the thyroid, bệnh cường giáp, remember so hyperthyroidism, bệnh cường giáp. When you have a benign tumor in your thyroid gland, is one of the reasons that you have the hyperthyroidism, because it increase the thyroid activities. Bệnh cường giáp, khi mà các bạn có một khối u lành tính ở tuyến giáp ý, thì nó sẽ tăng cường hoạt động của tuyến giáp và nó sẽ tiết ra quá nhiều hormone và sẽ dẫn đến các triệu chứng của bệnh. Và khi mà có một cái khối u lành tính ở trong tuyến giáp của các bạn, có nghĩa là các bạn những cái người mà có u giáp ấy thường sẽ đi theo là sẽ bị bệnh cường giáp, increase the level of hormone in the thyroid. That's why because they have a benign tumors and they work, they function like normal cells. So what about the malignant tumors? They can invade or spread to other parts of the bodies. They have the high rates of the division. They spread by forming the metastasis. They travel through the circulation system, and they're very difficult to treat. In this case, we have the rare cancer, right? We have a cancer cell when we have a malignant tumors. This could go to the metastasis. And here the frequency of certain tissues becomes cancerous, the cause of disease development, and efficacies of the treatment is variables. Not think about the types of cancer that is most common. We have some types of cancer like the breast cancer. Breast cancer is very common. Some kinds like the respiratory system, we have a lung cancers. In the digestive system, we have a colon cancers and skins also. So we have several types. It's very high cases of the genital cells. And think about this, is most of the cells in this tissue, they are replacing themselves all the time. So that's why they have the high chance to accumulate the mutations. So what about the heart cancer? The very, very low rate of the heart cancer. That's why. Do you have any reasons why it's very rare case of the heart cancers? Normally we don't have a heart cancer. For the heart cancer, most of the case of the heart cancer is just about we have the metastasis from other organ to travel to the heart cancer, and they have the tumor there. With no very, very rare case that we have the cancer cell that's originating from the heart cell. So the heart cancer is a very rare case here, and they in the soft tissue ung thư mô mềm, they include the heart tissue here, but we don't have the heart cancer because it's too low level. You know why? Why we don't have the heart cancer? Very rare case of the heart cancer.
Maybe because the heart muscle don't replicate that much.
Don't replicate much.
I think it's because they are differentiated.
They are differentiated.
Yeah.
All others are differentiated. Even in breast, in lungs, in the digestive system, we have a numbers of the cell that has been differentiated. If the cell is not differentiated, they don't know what job they are. Most of the cell in these kinds of organs are differentiated. Riêng biệt hóa đúng không? Em mới nhắc đến biệt hóa đúng không? Hầu hết tất cả các tế bào ở trong các bào quan trong các cơ quan khác thì nó đều phải đã biệt hóa rồi thì nó mới biết chức năng nhiệm vụ của nó là gì. Và trong tim cũng vậy, các tế bào tim thì đã rất biệt hóa rồi. Không biệt hóa thì nó không biết chức năng của nó là gì đâu.
Maybe because the cells of the heart is very complex. That's why it's hard
They are
to
complex.
divide.
They are more complex than other.
Yes, I think so.
Okay, all right. This is an idea. So, okay, think about the types of cancer that are most common. We have breast cancer, we have the digestive system, we have a skin, we have a lung cancers. They are the cell in this kinds of tissue. They are replacing themselves all the time. For example, the breast cancer tissue, they are constantly affected by the hormones, and they always growing and shrinking. And for the digestive system, they are continuously divide and replace every day. And the skin also, the skin beside they are divided rapidly, and it's easy for them to accumulate the mutations. They also has been exposed to the ultraviolet rays for the skins and carcinogens in foods in the colons. As a result, likelihood of mutation is high, and also these mutation will pass to the cell divisions during the cell or divisions, and the next descendant they have the mutation, they accumulate the mutation. In the end, we have the cancer. But for the heart cells, in contrast, they does not get exposed to many carcinogens. They do not expose to the toxic from food. For the liver cell, they do not expose to toxic foods, because the livers, they are detoxified organs, so they expose to the toxic all the time. For the digestive system, they expose to the toxic from the food all the time. From the lungs, they expose the toxic through the air all the times, like nicotine from the smoke, right? For the skin, they expose to the ultraviolet, the UV from the sunlight all the time. But the heart, first, they are do not expose to the many carcinogens, just only in the blood. And the heart is not often replicated. Remember about the cell cycles. When we have a cell cycle here, we have a G1, we have a S phase, we have a G2, and we have an M here. Remember that, as I said, some cells withdraw from the cell cycle to go to Z0. Because, and one of them, we have a neuron cell, right? We have neurons. I give you an example of neurons, muscles, and also the heart cell. Actually, we have a several kind of cell that jumps, that's withdraws from the cell cycles to jumps into the Z0. And the heart cell is one of them. When they do not divide a lot, That means there's no possibility for them to accumulate or to have the mutation, and no possibility to accumulate the mutations, right? And also the heart cells is made of connective tissues. The connective tissues, that their tissue is really connective. Like one student who said that they are more complicated is might be the idea. So connective. Connective tissue, and the master cell do not turn over very fast. That make them very resistant to become cancerous. So that's why we have a low rate of the heart cancer. And also with the brain, you have the brains here, but very is not quite high, but you have numbers of brain here. But I give you an example that the neuron cell is dumped into the synapse, right? But for the brain cancer, they most come from the pituitary glands, từ tuyến yên, or pineal gland, từ tuyến tùng. And most brain tumors come from the glial cells, tế bào thần kinh đệm, as most of them come from the glial cell. Glial cell. They come from the glial cells. It's not about the neuron cells. So they come from glial cell or other non-neuron cells. So that's why we still have the brain cancer and other nervous system, but it's not really high rate, right? So is the relation between the cell cycles and also the cancers. With cell is more divide, they are divided more rapidly, and they expose to the toxic, exposed to the carcinogens. We have the high possibility to get cancer from this organ. Okay. And next, about the cell cycle regulator and cancer. We have two types of cell cycle regulator may promote the development of cancer. First, for the positive regulators may be overactive. We have oncogenes, and I remember the CDKs plus cyclines. We also call them the positive regulator. And the next, the negative regulators, tumor suppressor may be inactivated. I give you some example like P53, P21s, or RB proteins. They are negative regulator. So they can enhance the positive and inactivate the negative. Okay. So the first case, when we have cancer, the positive regulators overactivate. The growth factors receptor may send the signal events, even the growth factor is not there. So we have overactivate. And the overactive form of these we call the oncogenes. While the numbers we call them the proto-oncogenes. Here, when the proto-oncogenes have mutations, they become oncogenes and we have a cancer. And this is one example. In the normal case, we have a rat. RAS is the G protein. Remember G proteins? We have a G proteins. RAS proteins. So in normal case, when they receive the growth factors, they will activate the cell communication, cell transduction, and cell response in cell divisions and cell proliferations. Whenever they receive no growth factors, RAS is off and no division or proliferations. But when they have mutations and they become the oncogenes, they are pro-oncogene here and we have oncogene here. They receive no growth factors, but because they have the problems, they always in the on stage. So they put the cells divide and unstoppables, and they lead to cancer. That's why when they overactive the positive regulators, we have cancer. And about the negative negative regulators, we have the P53. Remember P53? I have so many time reminding you about the P53s. When we have the cell damage, we are firstly they increase the level of P53s. They are negative or cell cycle regulators, and they will stop the cell at the G1 stage. They stop the cell at the checkpoint. The cell cannot pass through the checkpoint, and or they can create the CDK inhibitors to inhibit the positive regulators, and then they increase the levels of DNA repairs enzymes. And if the DNA cannot is fixable, unfixable, they will trigger the cells under the apoptosis. So if we have a mutation in the P53, 53s now become non-functions. And when we have a DNA damage, the DNA cannot repair because nothing stopped them at the checkpoint, nothing increase the level of DNA repairs. So we have a mutation. Remutation will pass through the next generations, and then we have a cancer. So that is two steps that will lead to the cancers. And now I show you videos about the cancers, and you can see that's how the cell can form the tumors, how they can coat the blood vessel inside the tumors, and how the cancer cell can pass through the bloodstream, travel around your bodies, and stay there. And when we have the metastasis, okay.
The human body is a complex and wondrous living machine. Like any machine, our bodies are composed of smaller parts that work together. The various tasks that are performed each day, such as breathing, eating and digesting food, and the movement of our blood, are performed by specialized structures known as organs. Examples of organs include the lungs, stomach, heart, and brain. All of these organs and other body parts like our skin and muscles are made of smaller living units called cells. Our lungs are a good example of how many cells work together to perform a specialized task. The cells in our lungs work together to allow us to take in oxygen from the air and get rid of waste products such as carbon dioxide. Our lungs are composed of many millions of cells working together to accomplish this task. Cancer is the result of a long process that begins when one of the cells in an organ or tissue becomes damaged or altered in a way that causes it to break free from the normal controls that allow our cells to work together in harmony. A group of misbehaving cells can cause the same kinds of problems in a body that a defective part would cause in any other type of machine. A normal cell will divide only when it receives a chemical signal telling it to do so. These signals are interpreted in the nucleus, and the cells reproduce their genetic information and divide into two identical daughter cells through a process called mitosis. Cancer cells do not obey this rule and will divide even if they do not receive appropriate signals. In addition to the signals that normal cells receive telling them to divide, they are also told when to stop dividing. This prevents too many cells from being made. In fact, the cell division process is a highly ordered process. This is a critical issue in cancer because cancer cells do not obey or require normal signals for division. This can lead to the formation of a mass of cells that piles up and may form a tumor. Also different from normal cells is the ability of cancer cells to continue dividing indefinitely. An important point about cells is that no matter what their job is in the body, they all have the same general structure. The cells that make up our lungs, heart, or brain are all similar in their overall appearance, even though their jobs are quite different. Just as the organs that they form are made of smaller structures, cells themselves are composed of smaller parts that help them perform their jobs. These smaller structures are called organelles. Of particular importance in cancer is the organelle known as the nucleus. The nucleus can be thought of as the brains of a cell. It contains the information that acts as the blueprint for each and every one of us, just as a manual would contain instructions to assemble a chair. Specifically, this information is contained within the chromosomes that reside in the nucleus. Individual units of information are called genes. At a chemical level, genes are made of deoxyribonucleic acid, or DNA. All of our cells contain the same set of information. It is only how the information is used that makes them different. For example, cells in the lungs use different bits of the blueprint to do their job than the cells in our stomach. In cancer, changes to the DNA cause some of the genes to fail to perform or to do their job in a way that causes problems for the affected individual. In short, all cancers are thought to result from changes to DNA that alter critical genes and change the behavior of the affected cells. If a change occurs to the nucleotide sequence, it is like having the letters of a word changed. An alteration in a gene is called a mutation. How do all these changes occur? There are actually a number of different things that can cause mutations. Examples include chemicals that can be swallowed or inhaled, such as those found in chewing tobacco and cigarette smoke, and radiation from the sun or artificial sources like a tanning bed. Sometimes mutations occur without any known external cause. They just happen. Certain genes make products that lead cells to reproduce. This would be equivalent to the gasoline system in cars. The genes that are responsible for making cells divide are known as proto-oncogenes. Changes in these normal genes lead to the production of oncogenes. The result may be cells that divide in the absence of proper signals. It is the equivalent of a gas pedal that is stuck in the on position, making a car go even when no foot is pushing down. Genes whose products function as the equivalent of cellular brakes also exist. As a group, these genes are known as tumor suppressors. Humans have 2 copies of each gene, one inherited from each parent. If a single copy of a tumor suppressor is damaged, the other copy is usually able to stop the cell from behaving abnormally. This would be like losing either the front or rear brakes of a car. The car may be damaged, but would still be able to stop. But if the second set of brakes is damaged as well, the car would not be able to stop, just as the cell would not be able to stop dividing if both copies of the tumor suppressor genes are damaged. The process by which tumors cause the body to provide them with nutrients is known as angiogenesis. Like the hungry plant in the Little Shop of Horrors, a growing tumor sends out signals that essentially say, "Feed me." The messages from the tumors cause nearby blood vessels to send over new extensions that deliver food and oxygen. Importantly, the blood vessels also serve as a passageway for the movement of tumor cells to neighboring and distant parts of the body. Spread of tumors to distant locations is of great importance in cancer. About 90% of the deaths due to cancer involve tumors that have spread around the body. The movement of tumor cells to other parts of the body is known as metastasis. Metastasis is a complex process during which cancer cells break off of the original or primary tumor and move through the body to form tumors at new locations. From the point of view of a cancer cell, this is a dangerous and often unsuccessful process. A trip through the body is full of hazards that cause the death of most cells that begin the journey, even tough cancer cells. To begin the process, individual cells must break away from the tumor and invade nearby vessels. The cells crawl along the surface of other cells and the fibrous, stringy structure surrounding them and then force their way in. Shown here is the invasion of the blood supply. Once inside a blood vessel, the cancer cells may perish from a variety of causes. Some cells die simply because they are unable to survive floating around in the bloodstream. Others may become damaged and die when they squeeze through tight spaces or bump into the walls of the blood vessels. Still, other migrating cells may be recognized and destroyed by cells of the immune system. How and where the migrating cells stop is different for different cancer types. Once the tumor cells are no longer moving, they can begin the process of forming a new tumor by leaving the blood vessel and beginning to reproduce in the new location. This does not always occur, and cells that have made it this far may still die or fail to divide. If the new environment is suitable, the newly arrived cell will begin to grow and a new tumor will develop. One way that the development of cancer is prevented is via the death of defective cells. A cell that becomes mutated or damaged will first attempt to repair the damage. If that is not possible, the cells will commit the cellular version of suicide. An orderly process called apoptosis leads to the breakdown of key cell parts and the death of the cell. Cancer cells lose this critical capability and will continue to divide. This can lead to the accumulation of cells that can become more and more abnormal. Due to the high rate at which cancer cells accumulate mutations, a tumor that originally started as a single abnormal cell is actually made up of many slightly different cells. They are all cancer cells and are similar to each other, but they may not all have the same sensitivity to any particular cancer drug or treatment. When this mixed bag of cells is exposed to a drug, most of the cells will probably die. Those cancer cells that are resistant to the treatment may reproduce to reform a new tumor. Importantly, this new tumor will be unlikely to respond to the same treatment. Their mutations make them invulnerable to that drug.
So it's all about these cancer cells, and now I will give you additional information about the stem cells. So for the girl in this class, when you have ever used the cosmetic that's containing the stem cells, have you ever seen or have you ever heard about the cosmetics that have the stem cells? Stem cell, tế bào gốc ạ. Nào các bạn nữ có bạn nào đang dùng hoặc là thấy mẹ mình dùng hay là thấy trên thị trường bán rất nhiều các sản phẩm mỹ phẩm mà ghi là tế bào gốc?
Yes.
Do you think these kinds of products contain the stem cell inside? So let's discover what is the stem cells. So the stem cells, they are the cells with potential to develop into many different types of cells in the bodies. We can call them the blank cells, or it's like a papers. There are no jobs, no work, nothing inside. And now you can write down, okay, you must become a liver cells. You need to become differentiated into kidney cells. And then we become what cell you want. That is stem cell, tế bào gốc. Cells, stem cells, the blank cells, unspecialized cells, they have the abilities of the proliferation. They can renew themselves for long periods of time, and they have the abilities of differentiation, biệt hóa. They have a potential to give rise to specialized cell types, and they have a plasticity. That is mean that they can differentiate in any type of cells you want. So here is one stem cells. They can differentiate into muscle cell, fat cells, bone cell, blood cell, nervous cell, etc. We have our classification of the stem cells is based on the sort of origins. We have the embryonic stem cells and we have adult stem cells. And for the different action and potential, is not all the stem cell can differentiate into any type of cells. We have a totipotent cells, tế bào gốc tổng năng. So in this cell type, it can differentiate into your whole bodies. It is strongest, powerful stem cells. And pluripotent, tế bào gốc toàn năng, they can differentiate in any cell type in your bodies, but it's very difficult to make a complete bodies. Multipotent, they can differentiate in several types of cells. For example, the hematopoietic stem cell. That is, stem cell can produce the blood cells, so they can produce the RB or red blood cell, white blood cells, and platelets. So they are multipotent. And the last, unipotent, is mean that they can differentiate in only one type of cells. For example, for the skin stem cell can only differentiate it into the skin cell. And the stem cells can divide slowly. So the first stem cells can renew themselves and they can provide a progenitor. Progenitors, if the cells can unspecialize, then they have a capability of undergoing the subdivisions and specialize, give rise on the additional stem cells, specialized cells, and they can give another type of the cells. For stem cell divisions, we have two types. For example, here we have a hematopoietic stem cell. That stem cell can produce the blood. They are blood making stem cells. They are blood making stem cell. For example, in this case, when they divide into one stem cell for the storage, and another cell is the specialized cell, just one red blood cell. In these kinds of divisions, we call them the obligate, asymmetric replication. And another case, for example, here we have a glial progenitor cells. The neuron stem cell here can specialize into two differentiated neuron cells, but one stem cell happen differentiated into daughter cells. Another stem cells we will notice. We have another stem cell here. They will notice and make up for the loss of the original stem cells by undergoing the mitosis, and they produce another two stem cells. It is stem cell, because they know that these stem cells have been produced two specialized cells, so they need to divide themselves to maintain the numbers of the stem cells. And this process we call the stoterics, differentiation. This is two ways of the cell divides. And the sources of the stem cell, we can take them from the cord, blood cell, umbilical cell, cord, and from the body cell, maybe we want extracts from the bone marrows. They have numerous of hematopoietic stem cells, or we can take them from the IVF, in vitro fertilizations, tinh nhân tạo. When we have the when we have the embryos, this means we have a chance to take the stem cell there, like when the eggs and the sperm mix together, we may have a zygote, right? A zygote, and they through the mitotics, they increase the numbers of the cells inside the zygote, and by several types of cell divisions, they will make, what happened here? They will make here a blastocyte, and inside the blastocyte, you have the inner cell mass we call the embryonic. And they are stem cells. Okay, we take the stem cells, and also we have the human therapeutics cloning. When we take the somatic stem cells and we insert into the nuclear a nucleated donor oocytes, and when we transfer this nuclear here into the nucleated donor oocytes, we can generate the embryonic stem cell also. And next, we have a huge discovery. This is huge discoveries of humans. Huge discovery. Once we can introduce new specific genes into already specialized somatic genes. We take the somatic genes, we introduce some reprogramming factors. We introduce a specific gene here. And then these kinds of genes may forget what types are these. They forget who they are, and they revert back into the stem cell. So it's huge discovery. So we have a very high source of the stem cell to do a research. So to beginnings, when the human discovered the stem cells, we only saw that's from the embryos that is raising the problem of ethics in science, cái vấn đề đạo đức sinh học. Because one team said that's okay, they are embryonic. This means that it can be a human, so you cannot do a research on the embryos. So through the times, we can discover so many sources of the stem cell to do a research. And for the induced pluripotent stem cell, here is a huge discoveries for the researcher who want to perform the research on the stem cell. And the purpose of the research the stem cell is because the cell variable potency that can cell renews. So in normal case, we can use for organ maintenance or repair the organs, but for the therapeutics, they can repair the damaged organ by introducing or injecting the extra stem cells. Injecting the extra stem cell into the damaged organs, and it's worked for hematopoietic stem cells and the skin cells. And with the Donald Thomas, he received this Nobel Prize in 1990s because he's the first successful hematopoietic stem cell transplantations in treatment of leukemia. Remember, do you know the leukemia? Bệnh máu trắng, bệnh bạch cầu, leukemias. When inside your bone marrows, you have a numerous of, in normal case, they are hematopoietic stem cell here, and they can induce numerous of blood types, blood cell type. But in some case, why one type of blood cell is increased too much, is causing the leukemias. But how we can treatment? We can take the hematopoietic stem cells in donor tomat. They take from the twins of the patients, and they introduce the hematopoietic stem cell into the bone marrows of the patients and can curb the disease. So he received the Nobel Prize. Here I have some videos about the stem cells, but maybe we don't have a time, so you can see at home about what is stem cells and what is the application of the stem cells. And now you can have attending checks, but during I can send you a link of the attending checks now. Response setting. Send the link in the chat first.
Link in the box chat.
Here. And I have a questions. Sorry, I sent to once. Do you know that Wix? I already sent you a link. And do you know this kinds of the cancer cell can become metastasis? Can generate.
Professor, there's an issue.
Professor, the link is a little.
Your name.
In the
Your
name,
name,
what's
your
your
ID.
name? It's a choice
125.
125. So
You
Oh,
just
sorry.
remake the attendance
Okay,
checks.
okay. Sorry, I will change. This work now. What's ID on scale? Okay, so what's his name here? So I have a question concerning the cancer cells. Do you think this width or types of cell can become the malignant cancer cell?
Teacher,
Yes.
the ID still is the
some
multiple
mistakes.
choice.
Yeah.
Okay, sorry.
Yeah.
A lot of mistake here. What's your name? What's your idea? Okay. It's not answer. Okay, this work now. Oh my God, there's one student choose their names by the numbers. So please, 4 students. I don't know why you choose 3 and 5, but please retake the Google form, okay?
Teacher, what do you mean option 1?
I think there is the mistake also. I would delete this. Okay, so it's work now. Name, IDs, okay. So I have a question, my last question. Do you think which types of cell can become the Mach-Lennon's cancer cells?
I think any cell that that is capable of mitosis should be capable of becoming a cancer cell.
No, it's not with you.
No. Okay.
No.
Nice.
Any cells have a capillary of the divisions could become the cancer cells, but not all of them can be the Mach-Lennon cell.
I think those cells need to be close to blood vessels, and it has
Close to the blood vessel.
yes, and
You know
they
that the blood vessel is everywhere in your bodies. There's no area that's in your body have no blood vessel.
Maybe epithelial cells.
Epithelial cells, because they are divided more rapidly.
Yes.
It's not the case. So.
I
Okay,
think
what?
can I answer? I think that the cell with the mutant which make the cell won't be able to connect with other cell would be the Mach-Lennon cancer cell.
Not really. Any idea?
The stem cell.
The stem cell, which type of stem cell?
I don't know.
So okay, so that's why I have additional information about the stem cell. You're right. Only stem cells we call them these cancer stem cells. They are types of the progenitors. They can found in the most types of the cancers, but they generally present only 1 to 3% of the all cell in the tumors. But they are only cell ability to generate the Mach-Lennon cells and fuse to the growth of the cancer. And only them can travel around your bodies and can differentiate into another tumor in other organs. And at least they must be the multipotents. That's for the unipotent, the cells, the skin cells cannot grow in the livers, right? It should be the stem cell, but at least they are multipotents. By that, when they travel from the skin cell, they can travel into the livers and they can differentiate it into the liver cells. Okay, so that's why you study a little bit about stem cells. That's because only cancer stem cells can become the Mach-Lennon cell. So I'm at the end of the lesson today. Today I have very important meetings on at 1 p.m. So I cannot give you the summarizations for the lecture today. I'm very sorry. If you have any questions, you can tell me or you can ask in the tutorial class. Okay, so goodbye. See you next week.
Goodbye.
Teacher.
See you later.
Goodbye, teacher.
Goodbye, teacher.
Goodbye, teacher.
Bye, teacher.